Today we're going to talk about sensory transduction in the visual system. Sensory transduction is the process of converting a sensory stimulus into something that our nervous system can understand. And what our nervous system understands is changes in membrane potential. A change in membrane potential in a receptor cell in response to sensory stimulus is called a receptor potential. So let's first talk about how the receptor potential happens in the visual system. Our receptor cells in the visual system are called photoreceptors, and they come in two flavors, rods and cones. The rod is called a rod because it looks kind of like a rod, and the cone is called a cone because it looks kind of like a triangle or cone. The rods and cones, the photoreceptors, are located at the very back of the retina. So light has to pass through multiple layers of cells before it can get to the back of the retina and stimulate the rods or cones. It'll be really important for you to be able to differentiate the structure and function of these two photoreceptors, but for now we're going to focus on things that are the same for both rods and cones. The stimulus in the visual system is photons of light. In the dark, let's erase our photon here. In the dark, rods and cones have cation channels that are propped open. So let's draw these cation channels. It's the same in the rod and the cone. And these channels are being held open by cyclic GMP. Okay. So cyclic GMP is holding open all of these cation channels. That means that in the dark, we have a bunch of positive ions entering the rod or the cone. At the terminal of the rod and cone, we have a system very similar to a regular neuron. When the cell is depolarized, voltage-gated calcium channels will open, and calcium will facilitate vesicle fusion, and rods and cones will release glutamate. So in the dark, if you record from a rod or cone, you would be recording a depolarization, and you would see that the rods and cones release glutamate. When we turn on the light, photons of light come in, and they will strike the photopigment. Rods and cones have photopigment, which is made up of a retinol plus an opsin. In rods, the photopigment is rhodopsin. Cones can have three different types of opsin. For now, we'll just call them cone opsins. When a photon of light hits the photopigment, it causes the retinol and the opsin to split. This will initiate a signal transduction cascade, which will ultimately lead to decreasing the amount of cyclic GMP in the photoreceptor. Because cyclic GMP is no longer present, these cation channels will close, and we won't have cations entering the cell. What that means is that in the light, the photoreceptor will be in a relatively hyperpolarized state, and we won't have any neurotransmitter release, or very little. So in the dark, photoreceptors are in a relatively depolarized state. They're releasing glutamate onto their downstream cells, which are called bipolar cells. Bipolar cells come in two flavors, an on bipolar cell, and an off bipolar cell. Cones can synapse with either on or off bipolar cells. Rods only synapse with on bipolar cells. On bipolar cells got their name because they turn on, that is, they're excited and release glutamate, in the light. Off bipolar cells, conversely, turn off, that is, they hyperpolarize in the light. First, let's see what happens in the dark. In the dark, the photoreceptors are releasing glutamate. The off bipolar cells express the AMPA receptor. As usual, when glutamate binds AMPA, the cation channel opens, potassium leaves, sodium enters. It causes a depolarization called an EPSP. However, the on bipolar cell expresses a receptor called mGluR6. This is a metabotropic receptor for glutamate. That means that when glutamate binds, this is a G protein coupled receptor and also a 7 transmembrane domain receptor. So 1, 2, 3, 4, 5, 6, 7. All right, here's our membrane. So glutamate binds. The first thing that happens is a conformational change to expose the G protein binding site. The G protein can then bind and be activated. That means that the GDP is replaced with a GTP. When that happens, the alpha subunit can split from the beta gamma subunit. The alpha subunit can then go open a nearby channel permeable to potassium. Potassium will then leave the cell. Positive ion leaving the cell will cause a hyperpolarization. So when glutamate binds mGluR6, the on bipolar cell is hyperpolarized. It's not going to be releasing glutamate. The off bipolar cell, because it expresses AMPA, will be depolarized by glutamate, and it will release glutamate itself. When we turn on the lights, the photoreceptors are no longer depolarized. They're actually in a relatively hyperpolarized state, which is the receptor potential, the change in membrane potential in response to light. Receptor potential. And they're not going to be releasing much glutamate onto their downstream bipolar cells. Remember that in the dark, the bipolar cell that was the off bipolar cell was being depolarized because AMPA receptors were open. Now, the AMPA receptors are going to close their cation channel, which means that the off bipolar cell no longer has positive ions entering. It's going to be in a relatively hyperpolarized state. Again, it's called the off bipolar cell because it's off, hyperpolarized, in the light. The on bipolar cell, however, is going to be released from this inhibition. This potassium channel is going to close, will no longer have positive ions leaving the cell. That means that the on bipolar cell is in a relatively depolarized state, and it will then release glutamate from its terminal. The on and off bipolar cells help to serve two different functions in our understanding of brightness. The on bipolar cell helps us perceive information about a light object on a dark background. While the opposite is true for the off bipolar cell. The bipolar cells have their synaptic contact with a cell called a retinal ganglion cell. Retinal ganglion cell. The retinal ganglion cells are the most typical neurons of the retina. Their axons form the optic nerve, which is cranial nerve number 2. Retinal ganglion cells, again, we can imagine them as our quintessential typical neuron. They all express the AMPA receptor. The retinal ganglion cell's response to light depends on if it's receiving input from an on bipolar cell or an off bipolar cell. Retinal ganglion cells that receive their information from an on bipolar cell are called on retinal ganglion cells, sometimes also called on center, off surround retinal ganglion cells, which we'll talk more about in class. Retinal ganglion cells that get their input from an off bipolar cell are called off retinal ganglion cells, or sometimes off center on surround. In the dark, the on bipolar cell is not releasing glutamate. That means that the on center off surround retinal ganglion cell is going to be in a relatively hyperpolarized state, and it is not going to be firing action potentials in its axon. The off center on surround retinal ganglion cell is receiving glutamate from the off bipolar cell. Its AMPA receptors will be bound by glutamate, their channels will open, and they will experience an EPSP. If the EPSP is sufficient to reach threshold, the off retinal ganglion cells will fire action potentials in their axons. When we turn on the light, now the on bipolar cell is releasing glutamate. That means glutamate will bind AMPA receptors on the on center off surround retinal ganglion cell, causing a depolarization called an EPSP. If that's sufficient to reach threshold, the on retinal ganglion cell will fire action potentials. In the light, the off bipolar cell is not releasing glutamate. That means AMPA on the off center on surround retinal ganglion cell is not going to be active. We're not going to have an EPSP. We're going to be in a relatively hyperpolarized state, and we're not going to be firing action potentials in the axon.